Good afternoon, students. Today we will be discussing the second lecture that is study of genetics of qualitative and quantitative characters. And as I told you in the previous lecture also, your subject name is crop improvement. When we say crop improvement, that means there is to be done improvement in the crop, and when we say for the improvement, that improvement is all about plant breeding. Improvement is all about plant breeding. And in plant breeding, we have to utilize different techniques, different processes, so that at the end, a crop could be improved. So our today's lecture is study of genetics of qualitative and quantitative characters. So before I actually come to the genetics of qualitative and quantitative characters, let's discuss or go through a bit of its introduction. So what is actually meant by the what is actually the requirement of plant breeding? If you call for the plant breeding, actually there are two basic requirements for the plant breeding, because crop improvement is all about plant breeding only. uh, when we say like what is to be done in the plant breeding, then plant breeding is having two requirements. Very first is, see, when improvement is going to take place, when there is certain genetic variation. When there is genetic variation means if we have this plant, until unless certain genes in it are not changed, say I have changed this one portion, genes of this much portion, then it is different from the plant that it was already existing, right? So for the improvement of the crop, what is required for the improvement of the crop? The basic thing that is required, that there must be the presence of genetic variation. And secondly, utilization of the variation during selection. Whatever variation we have induced, we are to select that variation, and after selection, variation is to be utilized for the purposes, for the particular purpose for which variation has been done. That means if we say, like, if any crop is to be improved, then the very basic criteria for the improvement of any particular crop, there is a requirement, like there must be the presence of genetic variation. If there is no genetic variation, then genetic variation has to be created, and there are number of methods by which we could go for the creation of the creation of the genetic variation. Like we can go for the hybridization, we can go for the mutation, and we can also go for the polyploidy. So all I discussed is like how we, if like your subject name is crop improvement, then how you can go for the crop improvement? Crop can only be improved if its genetic content is altered, right? And genetic content, how it can be altered? If there exist certain variations in the genetic content. Variation, say if we have a gene, if we have a chromosome, and the here, this is a sequence, this was the normal genetic content that was present in this particular plant. But when variations were induced, say here is T and here got the placement of C, this is the variation in the genetic content, that is said to be genetic variation. So if any crop is to be improved, we are to look after, we are to work on its genetic content, and there must be the variation in the genetic content. Secondly, it is not like when we were going for the genetic variation, then say this is the required variation that was to exist, but say this does not exist, what variation took place? G, C, and here came the placement of A and T. This variation also took place and this variation also took place. But we were looking after the presence of this variation only. So once variation has taken place, that variation is to be selected. like two variations have taken place, we need to decide which one variation is to be selected. So this when number of variations are taking place, then the very second step in the crop improvement is the selection. If you selected the right genetic variation, it will give you the desirable result, but if you selected the wrong genetic variation, it won't be giving you the desirable results. So once variation has taken place, selection is to be done, and whatever selection is done, this selection is to be utilized for the purpose required. This is what normally genetic variation is taking place. But what to do under the circumstances when when variation is no more taking place naturally? In that cases, if variation is no more taking place normally, then what we should do? Then we are to create the variation, and the variation can be created by three methods: hybridization, mutation, and polyploidy. How crop improvement is actually involving genetic variation. Genetic variation means change in the gene. So when we have, see, we in the hybridization we have parent 1 that is crossed with the parent 2, right? And then we have a hybrid. Some genes from this parent are transferred into the hybrid, and some genes from this parent are transferred into the hybrid. So this hybrid is having different genetic variations, different genetic content, where some of the genetic content is the result of parent 1 and another is the result of parent 2. So this, in this way also genetic variation can be created. Secondly, you can go for the mutation. Mutation is basically of two types. Very first, if it is taking place naturally, then it is okay. Otherwise, you need to go for the induced mutation. And in induced mutation, you can go either for the treatment with certain chemicals, or you can go for the physical treatment. When we say chemicals, then you are to treat your DNA with certain chemicals. And when we say physical, physically it is to be induced, then you can expose the genetic content in the presence of say UV radiations and so on, right? So this is the basic introduction, like what is how improvement can be done, and once improvement is being done, then how improvement is to be selected and how genetic variation is to be utilized. That means we can very easily say, like, selection is the second basic step that is required in the crop improvement. Again and again I'm stressing, like, crop improvement is all about plant breeding, because in plant breeding also we are learning about the different technologies that is required for the improvement of the crop. So whenever we are going for the plant breeding, or whether I say it is crop improvement, then very first step in the crop improvement is genetic variation, and the second step is all about selection. What is to be selection? Like I gave you two, I gave you normal. In the previous slide I told you there is normal gene sequence, then I told you gene sequence that is desirable, and then I told you the genetic variation has taken place in a manner that the gene sequence is no more desirable, it is not desirable. And we were to do, we were to go for the selection of only desirable genotype. Here I discussed only two genotype, two gene sequences. But if we talk for a population where we have number of genetic variations, then what actually selection is involving? What does actually selection does? It actually involves the identification and isolation of desirable plants from a variable population. So this is all variable population, and from this variable population, the plants which have desired genotype, they are to be identified, and later on they are to be isolated from this crowd. That is identification and isolation. And you must be thinking now how identification is to be done or how selection is to be done. It can be done on the basis of phenotype. Say your desirable genotypes give you 5 feet height of the plant, so you can easily identify the plants with 5 feet. They can be easily on the basis of phenotype, that is physical appearance. they can be easily identified and later on they can be selected. That means selection is based on phenotype. But when we talk for any character, this was all about improvement of the crop. But do you know what actually is meant by the character and how character gets developed? Character means any trait. We can say that trait can be either height, it can be either shape, anything. Now, when a character develops, development of the character is always regulated by either one gene or it is regulated by either more than one gene, that is by a group of genes. And in the genetics, what characters are there? Those characters are broadly divided into two categories. Very first is qualitative and another is quantitative. So all I have discussed is, first I told you the basic requirement of plant breeding. Then I told you if genetic variation is present then it is good. If it is not present then it is to be induced. Then I told you selection is the second method that is to be utilized for the crop improvement. And finally I came to your topic, that is character. Basically we have to talk for the two kinds of characters. Very first is qualitative and another is quantitative. But before I talk for the qualitative, I would like to introduce like character is the one trait that is actually regulated by either one gene or either by the group of genes. And broadly traits are divided into the two categories on the basis of the phenotype. Very first is qualitative and another is quantitative. Now we come back to our topic, that is genetics of qualitative and quantitative characters. But before I enter into the depth of genetics, I must make you all familiar with what are actually qualitative and quantitative characters. So character is what? Character is any morphological, anatomical, or biochemical feature of an organism. And it is very rare, like character is affected by the genetic background or by the environment. Generally if we talk for the expression of the character, then character is generally governed by either one gene or either by the few genes. Once it is governed by the one or few genes, they also gives you the easily detectable effects. And the genes which are actually regulating the expression of the characters, these genes are said to be oligogenes. And the characters that is produced by these oligogenes, they show different classes. And this is the reason this is known as qualitative characters. Qualitative means on the basis of quality, characteristics are divided or they are kept into different classes. When there is a distinction among the classes, we call it as the qualitative characters. But on the other hand, development of the, or say expression of the character is affected by the genetic background, the environment. If it is not affected by the genetic background, the environment, then in that case it was being governed by the oligogenes. But once it is being affected by the background, in that particular case expression of the character is governed by the several genes. And when we say several genes, we basically talk for the polygenes. And in these cases, and in this case whatever effects are produced, they are of additive nature. Like one effect will affect the another effect will affect certain another effect. And in this way it is governed by several genes which are said to be polygenes. Now the characters which are produced by these polygenes, they are said to be quantitative characters, because actually it is affected by the environment. And effect can be smaller, effect can be in the medium range, or effect can be at a very severe stage. So that means the quantity of the expression is varying as a result of the effect of environment. So now The characters which are produced by these genes, they are said to be polygenes. But these polygenes, they do not show any clear cut policy, and they have to be studied by measurement. So wherever we talk for the measurement, and wherever there is no distinction in the classes, we call those characteristics as quantitative characteristics. So coming in detail about qualitative characters. So qualitative trait is any trait. That means it is a character can be called as a trait also. So qualitative trait is a character that fits into discrete categories. That means separate and distinct categories. Because when we talk for the qualities, on the basis of quality, we say if we say we are talking for the quality, say length. Then on the basis of length, we can keep the individual in one category. Then on the basis of say quality, we can keep it on the another category. That means on the basis of qualitative characteristics, we can place say different characters in the different categories. And when there is a distinction, when there is a distinction into discrete, then we call it as a qualitative characters. For example, see, if a species of plant had red leaves or yellow leaves and nothing in between, this would be a discrete trait. Now we know the qualities like leaves are either of red color or either yellow colors, and in between there is no category. So this is qualitative character. Yes or no traits. Traits where an organism either has a trait or does not has also fit into this category. That means a red color is present or not present. On that basis also you can fit a particular trait into the categories. And here single gene or a small group of genes are actually controlling the qualitative traits. Qualitative trait is also said to be a feature that is either present or not present, depending on whether the gene responsible for the trait is present. That means functional or non-functional. See, if I'm saying a red color of the leaves, now red color is present or red color is not present. This turn, this in turn is regulated by this in turn is regulated by a gene which is either functional or which is not functional. If the gene is functional, if I say the gene is functional, then a red color is present. If I say the gene is non-functional, then red color is absent. I hope it is clear now. So qualitative trait is a feature which is either present or not present. So here red color is either present or it is not present. And this in turn depends upon the gene that is responsible for this trait. So whatever gene is controlling the red color, if it is present, that means if it is functional, then red color is present. If it is non-functional, then red color is not present. Another example here that is given is a flower color. A flower color can be red, brown, green, or yellow. That means it is not all the time flower color is going to be red. Sometimes red color is present and sometimes it is not present. Similarly, for the brown, green, or yellow. And if we talk apart from the agriculture, then another example is blood group. That means certain individuals have A positive blood group, certain have B positive, then O, so and AB, right? So we say B positive or not. So B positive is present or not. This is said to be qualitative characteristics. See, this can also be considered as one of the example of qualitative characters where plants are of a longer height and plants are of shorter height. these two are of same category, that means color is same, but one is gaining very high height and another one is of shorter height. Similarly is the case here. So these are not as dark as these plants, so color is either present and color is either not present. So here two qualities, that means two traits are being discussed, one is height and another is color. This qualitative traits, that means I already discussed with you all, like qualitative traits are usually, when we talk for the qualitative characteristics, these are encoded by certain genes. Now if I say, as I given the previous example and we talked for the height, so this is qualitative trait, that means qualitative character, and this character in turn is regulated by either one gene, or either this character is regulated by more than one gene, more than one gene. And whatever this say, if I say this is we are talking for the trait, say height, but when we talk for the qualitative when we talk for the qualitative characteristics, then these qualitative characteristics are not affected by the environment. They have nothing to do, they are not going to give any response to the change in the environment. This can be better understood by an example. If a flower is genetically programmed to produce blue flowers, that means you have a flower and its genetic content is like, the genetic content of this flower is like it is always going to produce blue flowers. That means it will continue to produce blue flower no matter where and how they are grown. That means it is least affected by the environment. If say this flower is grown in the Amritsar, then also this is going to give you blue flower. If it is grown in the Nepal, then also it is going to give you blue flower only. That means the genetic content of the flower is of category that it is programmed to always give rise to it is programmed to always give rise to say blue flowers only, right? That means it has nothing to do with the changes in the environment. And its genetic content is programmed like whatever environmental circumstances it is given, it is always programmed to give rise to the blue flowers only. Then we now need to think like how these qualitative traits can be analyzed. These are the discrete values, and discrete values means they can be analyzed by counts and ratio. How many number of flowers, blue flowers are produced, that can be considered. For example, the number of blue flowers can be counted exactly, as I already said, because these are the discrete values, so they can be analyzed by counts and ratio. And how many blue flowers are produced per flower, that can be determined, right? So this is all about qualitative characters. Now we will talk for the quantitative characters. Quantitative characters means the characters that occur as a continuous range of variation. And if you remember in the beginning also I told you these are the characteristics which have influence of environment. That means here if I say we had a flower that is programmed, if that is programmed to produce blue flower, but when it is given area that is said to be Amritsar, then it is producing blue flower. But when it will be sown in the Nepal, it is no more going to give you blue flowers. And again, blue flower are going to be produced or not, this in turn is regulated by certain genes, which may be one or more in the number. But when we were talking for the qualitative, the kinds of gene were oligogenes, but here the kind of genes are polygenes. If there is existing continuous range of variation, that means when in Amritsar it will give you certain variation, But when it is sown in the Nepal, then the variation level can be less or it can be high as comparative to the plants plant when it was sown in the Amritsar, right? What could we conclude from here? We can conclude that traits occur over a range. These like traits occur over a range can be very easily understood by an example, that is length of a lizard tail. Now, length of the lizard tail, this is the tail, this is the lizard, say, then the length of the lizard tail, it can vary. It can vary means the length can be this much, it can be this much, and it can be this much. If it is this much, it can be kept into category say 1. If it is this much, then it can be kept into the category say 2. And if it is this much, then it can be kept into the category say 3. That means the trait is now divided into range, and this is what is said to be on the basis of quantity. How much long is the length of the tail? On that quantity basis, it is divided into the distinct classes, and this is the way it is said to be quantitative characteristics. And here, whether this much length is going to be present, or this much is going to be present, or this much is going to be present, this in turn is not controlled by a single gene. Instead, it is controlled by a larger group of the genes, and those larger group of the genes are said to be polygenes. That means quantitative trait is an attribute that falls on a continuum of series. For example, height of the plant, milk yield by the cattle, and obviously the values are not constant values because they are affected by the environment. So the values will keep on changing on the basis of what environmental conditions are given. See, this is the example of quantitative characters. How much we are talking here for the quantity. So these are determined by the large number of genes. These are under the influence of environment, and they spread over a range of values. More clearly, qualitative and quantitative characters can be understood by giving a difference among the two. So when we talk for the qualitative characteristics, then in qualitative characteristics we are no more representing any statistical data. But in the quantitative, we need to represent the statistical data. Data is of kind that can be observed and not measured, but here data can be measured accurately because we are talking for the quantity, so data can be measured accurately. It is considered to be subjective, whereas it is considered to be objective. It helps to understand the underlying reason, opinions, and motivation. But here it does not cover the measurable data, and that data is no more covered because it is to formulate the theories and facts and uncover patterns. Uses either hypothesis or research questions. Here the mainly use is hypothesis. This is semi-stretched or unstretched methods, and here data collection methods are highly structured. Population is small and the sample population is very large. So with this, I told you the significance of crop improvement. Then I told you what are the two basic requirements of the crop improvement. Then we discussed what are traits. Traits are divided into how many categories, and then we talked for the features of qualitative characteristics, and then we talked for the features of quantitative characteristics, right? Now we come to the actual topic, that is genetics of qualitative or and quantitative characteristics. The genetics is what? When we are talking for the genetic makeup, that is genetics. And in the genetics, what we actually talk? We actually talk for the inheritance. In inheritance means how the genes are being transferred from the one generation to the another. That is actually what we focus within the genetics. But now genetics of qualitative and quantitative characteristics is to be studied. So genetics of both qualitative and quantitative characters, it actually follows the laws of inheritance. And these laws of inheritance were in turn given by a scientist named Mendel. Now, what effects are going to be produced in terms of qualitative characters or in terms of quantitative characters? They are totally different in both magnitude and mode of action. Since the procedures that are to be utilized for the measurement of qualitative characters and the procedures which are to be measured for the say measurement of quantitative characters, they both differ from each other. And this is the reason various techniques are used to study the two types of characters are also different, because the effect of individual gene is different in both the cases. Say if I say for a single gene, say if say for the gene A, if I say for the gene A, then this gene A will give different effect in this qualitative characters, and it will give different characteristics on quantitative characteristics. That means the effect are totally different in both magnitude and mode of action. So various techniques are required so that we could study the two types of characters. The various techniques are also different. And it is universally accepted that the characters, whatever character we talk for, those characters are present on the gene, and genes are normally located onto the chromosome, and these chromosomes are itself present in the nuclei of the cell. Here is the presence of chromosome, and each chromosome is having an identical partner that is called its homologue. That is called its homologue. Now, when meiosis takes place, till now what I have told you, like a single trait is controlled by a gene. Gene is located onto the chromosome. This is a gene, then gene is located onto the chromosome. And chromosome is having a homologue, that means similar copy of it. Now, I will be talking for the process that is said to be meiosis. In meiosis, what happens? These two homologues actually separate. See, I'm making it a bit smaller. This is one homologue, and this is second homologue. These two homologues, they separate and then they move to the opposite pole. They separate and move to the opposite pole, means here this is the anaphase 1 where there occurs a separation. Then one is moving to the one pole of the cell, and another homologue moves to the another pole of the cell. That means here when it was diploid, now here it is haploid. Now this diploid nature can again be restored by the process that is said to be fertilization. That means if this again fertilizes with this, then again we can have the same number of chromosome, that is 2n, right? This is what I'm telling you, like trait is controlled by the gene, and where is that gene present, and where is that chromosome present within the nuclei. And during meiosis at the anaphase stage, what actually is taking place, that means two homologues of a chromosomes, they separate and then they move to the opposite poles, and this movement to the opposite poles, it takes place at first anaphase. Now, the gametes which are produced, they receive one of the two homologues. Either they received this homologue, either they received this homologue. But if we say like two are to be restored, then it can be done by the fertilization. Okay. Now, as I said, like there are different methods for studying the qualitative characters, and these are few that I will be discussing now. Very first is dominance. Dominance. See, this is dominance. Dominance is name itself is indicating like some thing is dominant in the nature, and dominant means it is no more allowing anything present near it, that means which is recessive, it will not allow the recessive trait to express itself. This is dominant. And don't forget what we are talking about, we are talking about genetics of genetics of qualitative traits. Genetics means how they are being inherited from one generation to another generation. So very first methodology that we are discussing is all about dominance. Dominance is what? Actually in the gene, say if this is the gene P that is controlling the purple color flower, it is having a homologue, as I've already discussed in the previous slide. This is homologue, and they can, this is a particular gene that is controlling the purple color of the flower, and we can call it as alleles also. The same the alternative forms of the gene, alternative forms of the same gene, they are said to be alleles, alternative forms. So when this purple flower that is having capital P genotype, it is controlled with the white flower that is having small p genotype. This is genotype PP, and this is phenotype. That means the color that is produced is purple color, and this is genotype and this is phenotype. What could be seen, that is phenotype, right? Now there is one thing with the one very special feature of the dominant, like whenever with the dominant there is the existence of recessive, it does not allow its expression. That means whenever from here one p will come, and from here small p will come. Now this is dominant and this is recessive. It is controlling a purple color and it controls the white color. But when this dominant is present along with the recessive, it masks the effect of this recessive gene, and as a result, the very first progeny, whatever flowers are produced in the very first progeny, they are all going to be of purple color. This is the inheritance of qualitative genes. That means it is the genetics of qualitative genes or qualitative characters. Say that means very first way to study the genetics is dominance, and dominance is the case where dominant gene is no more allowing the recessive gene to express its trait. This is the F1 generation where we had all the flowers of purple color only. But you know, when these purple flowers were crossed with the purple flowers only, Then in the second generation, we had the flowers. In the second generation, we had the flowers, and there, when this genotype came, then we also had one flower of white color. So this is all about the we can say this is all about the inheritance of qualitative characteristics, right? This is the example of this is the demonstration of dominance. Now the next that we will discuss is segregation and independent assortment. And this is also under inheritance or say genetics of qualitative characteristics. What is segregation? Segregation is two alleles of a gene. Say we are considering, I again consider the same gene that is giving purple color flower. Normally what happens during the meiosis, these two get separated and one moves to one end and another moves to the another end. But here law of segregation says these two alleles, they remain separate. That means they will remain separate, and after separation they are not going to contaminate with each other. That means after that they are not going to meet. And when any gamete is formed, like this, when that means these two genes will separate. Similarly for the another case, pp, that is producing the white flowers, the alleles of two genes, they get separated, and then there is the F1 generation. And in the F1 generation, see, there is no capital P, there is no small p. That means we had a different genotype. Okay, so the conclusion is somatic cell of individual has two copies of the genes. That means this is the somatic cell. This is the not somatic cell. This is the gamete produced. This is said to be gamete because it is having single copy of a gene. That means it is from the here are two alleles, but it is having single allele. Gamete is the one that is having single allele of any gene. But when it is duplicated, say in the F2, when we have capital P, capital B, small p, when it is having two copies of the allele, then we call it as a somatic cell. So I mainly want to tell you like what is the genetics of say qualitative characteristics. In the genetics we are to discuss inheritance, and this is the another law of inheritance that was given by the Mendel, where I discussed like in this Mendel's law, inheritance is the taking place in the way that out of the two alleles, they are not going to remain together. These two alleles separate, and then they give rise to the gamete and they give rise to the somatic cell. Then on the basis of the inheritance of these qualitative characteristics, now you must be thinking why it is qualitative characteristic. It is qualitative because either the color is purple or either the color is white. Purple color is present or it is not present. If it is present, that means the gene that is actually controlling the purple color is functional. And if purple color is not present, that means the gene actually controlling the purple color is no more functional. That means it is non-functional, and that is the reason it is giving rise to white. And again I repeat, here the expression is controlled by single gene or by a group of genes, and those genes are said to be oligogenes. On the basis of conclusion, whether the two alleles are present or whether the one allele is present, we call it as a gamete or we call it as a somatic cell, right? If we had two alleles of the gene, then we call it as a pair. But if we have, this is said to be hybrid. Now I said no, in the F1 generation hybrid is being produced. What is that hybrid? On the basis of genetic content, we call this hybrid as the one that is having dissimilar copies of a gene. See, dissimilar copies of the gene, this was one gene, this is another gene. But here there is a capital P and here it is small p. That means both the genes are dissimilar. But here when we say we have identical a set of the genes, and we call it as a somatic cell. This was also called as law of purity of gametes. Next is the law of independent assortment. See, once they got separated, it is not once the two alleles got separated. Now they can independently. This can independently mate with this one, and this can independently segregate, means can get combined with this one. So segregation is independent to each other, right? This is said to be law of independent assortment. For example, I give you very easy example, like say there are 10 girls in your class. Now out of these 10 girls, say there are 5 groups. In each group we have 2 girls. Now it is not always like these 2 girls are going to remain together and these 2 girls are going to remain together. These girls can independently join this group, and these girls can independently join this group, or they can join this group. This is said to be independent assortment. One more example I can give you. Suppose if I say CR to sit on the first bench, but CR can sit on the second bench also, CR can sit on the third bench also. This is said to be independent assortment. That means where to sit and at what place to sit, that in turn is no more controlled. And this is when I talk this in terms of qualitative characteristics. Then in terms of qualitative characteristics, we say inheritance, like what character is to be separated and where it is to be segregated. This is independent, right? Next is pleiotropy, inheritance, or within the genetics of qualitative characters, we also study pleiotropy. Pleo means many. Here we say we have 3 genes, and this gene is giving phenotype 1, this gene gives phenotype 2, and this gene gives phenotype 3. This is monotropy. That means one phenotype is being controlled by one gene. Similarly here, phenotype 2 is being controlled by second genotype. Similarly here, phenotype 3 is controlled by third genotype. This is monotropy. But qualitative characteristics are, when we go through the genetics of qualitative characteristics, we come to know expression of one phenotype is also affected by more than one gene. That means that if phenotype 2 is to be produced, then it is not only being produced by gene 2, it is also produced by the combination of interaction of gene 1 and gene 3. That means gene, normally when we say one gene is controlling a single character only, like one gene affects a single character. But in many cases, one gene is also found to affect more than one character. It is affecting this character, that is fine, but it is also affecting the second character also. These genes which are affecting more than one character, we call it as a pleiotropic genes. And this condition where one gene is affecting more than one character, this condition is said to be pleiotropy. That means pleiotropy describes the genetic effect of a single gene on multiple pleiotropic. What we understand from the pleiotropic, we understand that single gene is having many phenotypic traits. This is pleiotropy, and in this way also we go for the study of genetics of qualitative traits. And the last feature that I will discuss is all about penetrance and expressivity. Penetrance is actually the percentage of individuals of a given genotype. It is actually the percentage of the individuals, those individuals which have a particular genotype, say A. But here penetrance means how deeply the character is penetrated, and expressivity means how that genotype, up to what extent that genotype is being expressed. Say this is the genotype, and say this genotype gives a red color. Now red color can be light red, it can be dark red, right? Now this is penetrance. Penetrance is what? Penetrance is percentage of individual with a given phenotype. But up to what extent this phenotype is being expressed, that is said to be expressivity. That means expressivity is calculated at the phenotype level. That means penetrance explains you how often the gene is expressed, like it is expressed once, twice, how often how regularly gene is expressed. But here we talk for the extent of gene expression. That means it is a quantitative measurement. Quantitative means how much genotype, how much quantity of the genotype is being expressed. But here we talk for quality, means red color is light or it is of dark color. That is quality. That means expressivity gives you the qualitative measurement. If the genotype is present and if the genotype is not present, on that basis, it explains whether the disease shows up. That means if disease is present, and on the basis of quality you can come to know how a disease is shown up. It describes the expression of a single gene. It gives the expression of a group of characters. If penetrance is incomplete, then it makes the genetic analysis more difficult. But the variable expressivity makes genetic analysis more difficult. Here these are the certain genes which when are developed, then they shows that there is a presence of cancer only in the gene. This is an example that is being dictated in terms of humans. Like if these two genes expression is taking place, then that particular person into which these genes will be present, they will be having cancer. But those individuals into which these genes are no more present, they are no more going to have any symptom of cancer. Here, characteristics of Marfan syndrome widely vary. Now, even if cancer is present, then at what level and in the different individuals, cancer is available up to what level, up to how much level, like whether it has reached the stage that it cannot be cured, whether it is at the stage it can be cured, or whether certain another effects are there. So on that basis, we call it as expressivity, right? With this, we complete the genetics of qualitative characters. Now, the last one, we are to talk for the inheritance of quantitative characters, or I can say genetics of quantitative characters. When we say genetics of quantitative characters, then in this case, already I have discussed here, the gene that is actually regulating is not the one, it is a number of the genes, and those genes are now called as oligogenes. Oligogenes are the genes that is giving the different variation, and on the basis of the different variations, it is permitting the classification of individuals into different classes, and those different classes are on the basis of characters concerned. Now, the characters which are governed by polygenes, they can also be called as a multiple factor, and but the ones which are showing discrete variations can also be called as quasi-quantitative characters. So this is all about the genetics of we can say this is all about the genetics of qualitative and quantitative characteristics, where I discussed with you all very big in the very beginning about plant breeding. Then I discussed the two basic requirement that is actually required for the crop improvement. Then we said what are characteristics. Then we called characteristics into, we defined characteristics into two categories. Then we learned in depth about qualitative characters. Then we learned in depth about quantitative characters. Later on, we said the since the effect of the two characters are different, so the methods used for its analysis are also different. In terms of qualitative, we said genetics can be studied by the methods like dominance, penetrance, and expressivity, uh, and segregation. Then segregation. Then we talked one more we talked for independent assortment. And then in terms of genetics of quantitative characters, we discussed multiple factor hypothesis. That means here the character is not only affected by a single environmental factor, it is affected by many number of genes which are said to be oligogenes. So this is all about your second lecture, that is genetics of qualitative and quantitative characteristics. If you have any doubt, you can discuss, or you can text, you can call, and whatever you feel convenient mode of communication, that you can in through that you can communicate with me and let me know your discrepancies. So thank you. This is all about today's lecture from my side. So stay home and stay blessed. Thank you.